thank you very much to Lauren for accepting the invitation and for this presentation. Lauren, she is currently a PhD candidate at Weill Cornell Medicine in New York under the supervision of Professor Imana Girasude. She has been working a lot with metagenomics, and I think this what she will talk about. She will tell us about, which is the microbiome in a bottle project. Um, Thank you very much, Lauren. Please, the stage is yours. Thank you.
Great. Thank you, Simone. Thank you for inviting me to give this talk about my work. I apologize in advance. My voice is a bit sore because I seem to have gotten sick along with everyone in New York, which is where I'm joining you from right now. Today, I will be telling you a story about a human gut microbiome dataset that I've been working on for basically my entire PhD. The analysis workflow I've developed to handle it, I'll only talk about that very briefly, um, and potentially some ideas that we could maybe work on together with respect to this data set. So this talk is maybe not in the same flavor as the others in the program. It's meant to be kind of a guided introduction to working with metagenomics data as someone in pangenomics, what to ask for, what to look out for, and to provide you with an overview to the gold standard benchmark that I've been working on for the last 5 years that I would love for you to work on as well if you're interested. So before I really dive into all of that, just as like a quick overview for those of us who are joining with maybe like a single mention of the microbiome during your undergrad. So microbiome refers to the community of organisms that inhabits a particular environment. And this environment can be host associated, it can be free living, just in some sort of abiotic situation. And for example, because we love focusing on human beings, the human microbiome consists of all the microbes, which means bacteria, fungi, viruses, archaea, among others, living on or inside the human body. And this includes environments like the gut, the skin, the mouth, and a bunch of other areas. So for example, I pulled this figure from Martino et al., and it shows how the human microbiome actually changes over time, over this person's entire lifespan, from the initial colonization of pioneer microbes starting at birth, all the way through the development of body site-specific microbial communities. So as this person matures, their microbiome increases in complexity till it reaches a relatively stable community structure at adulthood. And moving on, why do we care? So why do we care about microbiomes? I'm sure you've heard it as a buzzword, especially the last 5 years. It seems like everyone and their grandma's taking a probiotic. And it's really not, it's really like, and for good reason, because we've discovered that microbiomes are essential for maintaining the health and function of their host environments, whether that's in humans or in animals or just in the world around us. And they can play roles such as aiding the digestion, protecting against pathogens, cycling nutrients, and for human beings, influencing behavior, and in our environment, influencing ecosystem stability. So on the left, we have a schematic representation of the role of like the gut microbiome in health and disease, and we have some examples of inputs and outputs. So let's say we're really, really stressed. It's grant writing season. We're eating a lot of Hershey's bars. This is very American reference. We might have a lot of inflammation markers start to pop up because we've accidentally dosed our gut microbiome with a whole lot of stressors like sugar. And on the right, we have various examples of the deleterious anthropogenic impacts of human beings on plant growth and soil health. And this is more of like a link into industrial agriculture. we have that. We have the impacts in white, followed by, in the yellow boxes, all the different ways that soil microbiomes can potentially be bioengineered to ameliorate their effects. Moving on. So now that I've sort of like contextualized what it is and why we should be studying the microbiome, first things first. So how do we do this? The foundational, though by no means straightforward, as I will get into, step of most microbiome studies is finding out who's in our samples. So we've got two different methods up on the slide there explaining like how we can go about doing this. We can just take short reads, classify them, like just align them somehow against a reference database using tools like BLAST and Kraken. That's one way. However, short sequencing reads can be very non-identifiable in the sense that they can be very non-specific to certain species genomes. So one, we're dealing with a bit of a specificity issue. And the set of species we've identified are intrinsically biased to the ones we've already cultured or studied heavily. And I'm studying the human gut microbiome. This doesn't really apply to me. It's very well studied already. But let's say you were, for some reason, studying chemotrophs living on the edges of volcanoes. You are probably not going to have a lot of luck blasting your reads that you've discovered on the edge of I'm struggling to name an active volcano, but yeah, to discover species living there that we already know of. So, attempt two. If we want to find novel unknown species, one thing we can do is try to infer metagenome assembled genomes, which are clusters of sequence material that share similar sequence or co-abundance properties. So MAGs can be thought of as our best possible reconstructions of the actual isolate genomes that comprise that original community from fragmented sequence data. And if you want to sort of tie it back to concepts you've heard before, MAGs are analogous to the classical microbiology concept of isolates, so singular colonies on an agar plate, analogous to the phylogenetic concept of, depending on what resolution you're looking at, a species or a subspecies or a strain. And that is what that is the sort of unit of measurement I'm trying to work with now. So if you're trying to build a pangenome graph from metagenomic data, we want to start first from either reference genome or a MAG. So a little bit more information, a little more context about how we study the microbiome before we really dive into what I've actually worked on. So let's say we start with some sort of sample, the human gut microbiome sample. So after we generate a sequencing data set from it, we would take all of those short sequencing reads and we'd assemble them into contigs, which are then clustered into our metagenome assembled genomes, or MAGs, which are then graded as low or high quality depending on the presence or absence of predefined marker genes. If we think that we're studying a very well characterized already sample, we can try to classify it against reference database, and hopefully, what we have found with our MAGs is representative of the original sample, though we don't actually know that because we don't have an answer key. And sort of the little asterisk that I want you all to take away from this slide is that every step in this reconstruction workflow is a source of error. And why is that a source of error? So before I dive into the exact details, I'll give you a bit of an analogy. So here we have some lovely colorful shapes that I'm going to break into 288 puzzle pieces. So let's say the challenge is to recover all of these shapes that comprise the original picture. So if I asked you to reconstruct every single one of these shapes, how many puzzle pieces would you need to do with? This is an information theory problem. Could you do with 10? What about 100? What if those puzzle pieces were smaller and they covered less than, let's say, 20% of each shape? what if some of these shapes had multiple colors in them? Let's say like this big orange triangle. What if it slowly gradated into red and it looked really similar to our red circles here? Well, ovals. How would you then solve this problem? That seems like a tough issue to overcome, and that's kind of the problem that we face when we look at a lot of metagenomic samples, especially samples taken from communities that are known to be really complex, like the human gut, like soil, et cetera, et cetera. So here's a visualization as to why the sequencing data to make reconstruction can give you an extremely biased picture of your original sample and potentially inaccurate and misleading. So because reads and contigs are short, the sequencing breadth and depth means that large portions of each isolate genome can be entirely unrepresented. So we've got some, let's say like we've got like the small shapes, like the square, the circle, the stars, and like the little flag shapes. Let's say they represent sequencing reads or contigs. The resolution at this point doesn't really matter. It just matters that it's a lot shorter than the genome. Let's say that the colored version, like the colored versions of those shapes are the ones that we actually recover in our sequencing experiment. All the ones in black completely missed, completely missed, because there's no way your sequencing experiment can possibly cover all of the genomic content of your sample. So let's say you have all of the red, the green, and the blue colored shapes to work with here. Let's say that going back 2 slides, after you get your sequencing reads, you try and assemble them into contigs, and then you try and cluster those contigs into mags. So the clustering process itself is obviously not perfect. So let's say we've clustered together most of the green stars and most of the green triangles together. We miss some of the sequencing content entirely, namely some of the triangles and some of the stars. And of course, all the stuff we didn't sequence. So the set of reconstructed mags may not actually be representative of the actual sample composition, which actually looks like this. Furthermore, we didn't know that we started out with 5 species. So mag inference itself is an unsupervised clustering problem, complex distribution of hidden variables, lots of missing data that we really, really want to observe. So that takes me to sort of finally getting to what I'm working on. And before that, I'm going to talk about some takeaways from my talk today. So if nothing else, I'd love to leave you with 3 big points to think of if you're ever considering working with metagenomics data, if I haven't already scared you off. So firstly, metagenomics data is inherently complex and procedurally contingent, which means that what you get is really dependent on what you do to your original sample. And what I do to my original sample not only includes the dry lab part of analyzing all the data, but also the wet lab part, the storage, the handling, the DNA extraction, the library prep, everything. Second issue is that even getting down to species identifications can be a really challenging endeavor. This, like what I've talked about in the previous slides, are like years and years of work. And the third point, which I will get to in the results section a little bit more, is that manual curation or careful visual analysis of your metagenomics data prevents downstream garbage in, garbage out issues for any sort of analysis you might want to do that starts with metagenomics data, which could include pangenomics. It can include functional genomics, where you're looking at the gene capacity, the phenotypic capacity of your entire sample, and so on and so forth. Which brings me to the project I've been working on, which is trying to address the fundamental unknowability problem of a metagenomic sample just right at the beginning. So what I want to do is I want to kind of do for the human gut microbiome what the Genome in a Bottle project has done for the human genome, at least before, telomere to telomere assemblies. So if you've heard of that, this is sort of the same idea. First of all, we want to make data, lots and lots and lots and lots and lots of data to comprehensively profile all of the sequence material in a single human gut microbiome. We're doing this with multiple DNA extraction methods. We're doing this with multiple technical replicates and sequencing technologies. And I'll go into that in a little bit. The second part is like we need to be able to integrate all of this data in a sane way, which is harder than you might expect. I spent a lot of time designing a workflow and analysis pipeline that integrates multiple technologies, lots of different types of sequencing data, each with their own strengths and weaknesses. And each type of sequencing data, fun fact, requires its own reconstruction tool. That's great. And which takes me to my goal, which is to create a quasi-gold standard benchmark data set of high confidence isolate genomes that fully comprises the healthy human gut microbiome it came from. So having all of these three things, the data, the workflow, and like the quote unquote answer key, will hopefully help us to benchmark future metagenomics tools, as well as provide a resource for lay people to study, for other researchers to study like these technologies from and what their strengths and weaknesses are. So the goal of the microbiome in the Bible project is high confidence, high confidence isolate genome or like high quality mag sets for the human gut microbiome samples, and obviously the matched data to go along with it. So let's say, again, we start with our human stool samples. We extract gut microbiome DNA using a few different methods. So I have listed here like 2 different DNA extraction methods because, and I will not go into this digression, each DNA extraction method will preferentially return DNA from certain types of organisms, and that's because the coding of each bacteria, whether or not it has a cell wall, affects how well it responds to some of these lysis methods, which means you will get more gram-positive bacteria, more gram-negative bacteria, depending on which extraction method you use. So it is obviously worth it to try a bunch of different extraction methods to try and get as comprehensive a view of our original sample as possible. And because we can't just stop with different extraction methods, we have to do multiple replicates to be able to compare and see how stable each replicate is. So we have 2 replicates of 2 different DNA extraction methods. And what we do is we make sequencing data from all of them. So this is the first part. This is the data that we've created. So this is our first goal. And let's say for each of these replicates, we will sequence them using multiple sequencing technologies. So I haven't repeated it just for the sake of visual acuity, but each replicate from each DNA extraction method undergoes Illumina sequencing and TELSeq synthetic long read sequencing. I will go a little bit into synthetic long reads in a bit, so just hold tight there. And then from each of these sequencing data sets, for each of these replicates, for each of these DNA extraction methods, I will try my best to reconstruct mags using them. And then these will function as input for merging into high quality sort of initial draft mags. So these are like these are the first pass guesses that I've come up with using metagenomics reconstruction methods. And then from there, taking our high quality initial draft mags, what we will do is we will integrate some more sequencing data, because for each of the replicates from each of the extraction methods, what I will do is I will sequence them. I have already sequenced them again using 3 different sequencing technologies: loop-seq linked read sequencing, or synthetic long reads, Oxford nanopore promethion long read sequencing, and some high-C sequencing as well. Then I'm going to integrate all of that sequencing data by mapping it to our high quality initial draft mags. And the little like tool symbol that I've indicated is the workflow that I've come up with. Like this has been under several drafts of revision, given like some of the preliminary data I've received. And then what we will do is we will take each cluster of sequencing material that was mapped to a single initial draft mag. So we'll take all of the Illumina reads that went into creating that mag. We'll take all the TelSeq reads that went into creating that mag. We'll take the LoopSeq, the Nanopore, and the Hi-C data that maps to that mag. And then we will do individual high quality single isolate reconstructions of all of those. And hopefully what that means is that from these clusters of sequencing material that we think have originated from a single isolate, we can reconstruct the full isolate genome from there. And that is the entire process of the microbiome in a bottle project. And the fun part is once we've got all of these isolate genomes, what we can do is we can compare them back to the original, each of the the technology-specific sequencing data sets that we had. And we can see, hey, okay, this is how much of the final metagenome assembled genome that Illumina was able to represent, and how much TelSeq was able to represent, how much Nanopore was able to represent, et cetera, et cetera, et cetera. So just as a corollary, we get a fun multi-technology head-to-head there as well, along with our final product. So before I start jumping into the data and the results, I'm going to sort of go over the pros and cons of currently available sequencing technologies. So I'm sure for a lot of you this is very, very old hat, but I'm just going to cover this really quickly for people who haven't especially worked with synthetic long reads before. So I want to provide some context about the sequencing technology we've included in this project. So thinking back to the puzzle pieces, we can sort of conceptualize short reads and long reads as like small and large puzzle pieces, and synthetic long reads or linked reads as sort of like an intermediate size. So synthetic long reads, if you think all the way back to the mid-2010s, the company 10x had synthetic long reads or linked reads or barcoded short reads. So what that means is like if you have a single genomic fragment, traditionally with library prep, you just like break it up into short reads and away we go, we sequence it. The step that you can add in during library prep and before sequencing is to take all the short reads associated with the same long read and assign a barcode to them in some way, shape, or form. 10x did this with oil droplets, and TelSeq and LoopSeq each do this in their own proprietary way. And what that means is that the barcode gives us sort of spatial information about each short read, indicating that a whole bunch of short reads are coming from the same genomic fragment. So within like X kilobase pairs they must be within X kilobase pairs with each other, which makes the assembly process easier and gives us sort of longer range insight into where each short read goes in a genome. So intermediate sized puzzle pieces. But in general, across all these technologies, whether it's Illumina or Nanopore or TelSeq or LoopSeq, the trade-off is generally between read length and read accuracy, what the throughput of the technology is, overall coverage, and the read error rate. One thing I didn't mention here is the cost. The Oxford Nanopore datasets definitely cost us a lot more money than the short read ones. And that should sort of hopefully contextualize how much data we've created, because we've managed to create some very, very large Oxford Nanopore PromethION data sets. So spoiler alert, this actually becomes a problem later on. So definitely keep a little asterisk next to that in your head. Conversely, the loop-seq data set that we generated, the synthetic long-read data set from Element Biosciences, is tiny. It's nearly 140x smaller than the equivalent short-read data set. So this is probably actually because of the library preparation procedure. So their synthetic long reads are generated from very, very high coverage of the original long genomic fragment. So that means that if I mentioned before that their accuracy is really high. Like these are incredibly like high quality long synthetic long reads, but an average of 2.5 kilo base pairs long. But that also means there isn't a lot of breadth-wise coverage of the species in the sample, which is why the hybrid technology workflow is structured in the way it is, because we can't actually recover MAGs from the loop-seq data set. So a small takeaway here, it's not just to show you numbers, is that not all sequencing technologies are created equal, and sort of as a bit of foreshadowing as to what each of the data sets are capable of and why they were integrated in the workflow and the way they are. So we're finally on data now. So let's say we've got our sequencing data sets. We've designed our workflow. We've created our hopefully high quality first pass metagenome assembled genomes. How do we find out if they are actually any good? So there's a few metrics that we can use and that hopefully will be of use to you if you ever decide to work with metagenomics data. So I've got a few metrics on the right: completeness, contamination, genome fraction, and proportion of unaligned bases. So I picked these out for specific reasons. So the completeness of a metagenome assembled genome is the proportion of single copy marker genes present in a MAG. Contamination is the average multiplicity of expected single copy marker genes. So contamination means let's say in a MAG there's like 5 copies of most of the marker genes, and then for 2 of them there are 10. What that means is that those 5 extra copies in each of those, in the last 2 single copy marker genes, are probably contamination. They might just be extra sequencing material, like more sequencing material from that same isolate, but they could also be sequence material from other species that was just similar enough to end up in that MAG. So they would be flagged as contamination. Completeness and contamination are proxies for genome fraction and the proportion of unaligned bases. Genome fraction being the proportion of a species reference genome in a MAG, and the proportion of unaligned bases meaning the proportion of the MAG that doesn't align to that reference genome. Why this is important is because sometimes what we'll be working with is a MAG that has never been seen before, and this happens a lot. This happens a lot. That is why there are whole reference databases of MAGs. There's just a whole lot of uncultured material in the world, and unless something can be cultured and like fully like whole genome sequence, it's not going to make it into a canonical reference database. So MAGs it is. So novel species are not something to shy away from. That's why we have somewhat non-reference based proxy metrics for working with MAGs. So on the x-axis, I have sets of MAGs reconstructed from each of our Illumina technical replicates, named for the DNA extraction method or the originating collaborator. So we have collaborators at Zymo, we have collaborators at Phase. So Zymo created 2 Illumina short-read sequencing datasets for us. Phase created 1. So you can see the median completeness of the Zymo technical replicates is not great. It's about 50. For a high quality MAG, you'd probably want about 90%. The Phase ones, however, did really well. And we can also see sort of like a similar trend with contamination. The Zymo technical replicates, they didn't have great contamination scores. It's 10, which means that 10% of the single copy marker genes had multiple copies in them. Phase did much better with a few outliers. So these are some of the metrics we can use to assess mag quality. And some of them are even reasonably good predictors of the original, of the reference-based genome fraction and proportion of unaligned bases. So just for fun, I've included two other potential metrics for the proportion of unaligned bases, the clade separation score and strain heterogeneity. The clade separation score detects contamination by evaluating whether sequences from different taxonomic clades are incorrectly binned together in the same mag. And the strain heterogeneity is the extent to which a mag contains multiple divergent copies of a marker gene that are within a certain amount of amino acid identity. So that's kind of like the key point here. So even if there are multiple unexpected copies of a single copy marker gene, that doesn't necessarily mean that the sequence content could still be pretty close to that of the mag species. And why I wanted to point that out is that the quality metric of interest, if you want to do some pangenomics on some mag data, is that strain heterogeneity is probably what you want to look at. So let's go to like a box whisker of strain heterogeneity for our 3 Illumina replicates and the mags we got from them. The mags you'd probably want to pull out for further pangenome analysis are the ones with really high strain heterogeneity scores. So these are between 80 to 100%. So what 80 to 100 means is that between 80 to 100% of the single copy marker genes in that mag have a multiplicity of copies in them that within a certain amino acid identity level. That means there's within the bounds of the species definition of sequence similarity, there are lots of copies of these single copy marker genes that exhibit a lot of variation in them. So aside from that, the reason also why I brought up completeness and contamination before is to sort of tie it back to the third takeaway that I mentioned earlier, which is that before actually taking on a pangenomic study of metagenomic data, have a very, very good look at what you what you want to start out with. So even after you've identified 4 mags of interest here, it is a really good idea to look at their completeness and their contamination afterwards. So the completeness is, as you can see, is pretty low. It's definitely under 90, which is not great, which means that this mag is probably pretty deeply incomplete. And it might be actually hard to pull out a reasonably, like the assembly graph comprising this mag might actually be really, really fragmented. And then the other, so like that's not ideal. However, the contamination, the proportion of unaligned bases tells you that most of the sequence material in this mag probably belongs to the species. So that is a good sign. So those are sort of the things that you might want to look at first pass. But I'm going to go straight back to more microbiome in a bottle project results right now, because if you see, like, all 4 of these have been classified, and that's generally true of the mags that we've discovered in the sense that most of the mags, the vast majority of the mags that we have discovered in the Illumina data set have been classified. So we have 97 mags in total. We discovered 70 species in total. And of those, only 5 out of the 71 classified mags. There was one repeat of a species in a single replicate. So in the phase data set, we discovered 2 mags that were classified as Mediterraneo-bacteriophagus. So there were 71 mags in total that were classified at the species level. Only 5 out of the 71 species occurred in 2 Illumina replicates, in more than one, which means that each of these technical replicates discovered a nearly private set of species. Luckily, these are all gut bacteria. Like I went back and checked against the database. These are all known to be gut species. And what this tells me is that the species that occurred in 2 replicates probably do exist. But what about the other 60-something species? Are they hallucinations? Are they chimeras? Are they chimeras in the sense that are the context comprised of sequence material from multiple species? Are they chimeras in the mag sense, in that the sequence content of contigs from different species were clustered in the same mags? Are they just a misclassified mag because the mag is too small? These are all things that I kind of want you to tuck into the back of your head as like a takeaway, that if you just have a single technical replicate of a single sample, yes, you can discover some like pretty high quality material in there. However, a lot of the not as high quality material can just be an artifact of the analysis process. And the other takeaway is that you need more corroboration by other sequencing technologies or other additional sequencing experiments. Species occurred in 2 Illumina replicates. This is the 71 mags that were classified at the species level. And the takeaway from all of this is that we need some more data, some more evidence to indicate that the other 60-something species actually does exist. So that's kind of what I've done with the Microbiome in a Bottle project. I have sequenced the same sample dozens of times. And one of those ways that I sequenced them was with TELSeq synthetic long-read sequencing. And I've also taken all of that data and made mags with them as well. So I discovered in total 70 species with the Illumina mags over 3 replicates. Over 4 replicates with TELSeq, I have discovered 79. So luckily, a majority of them overlap. 51 species overlap, which is great. But that's only a slim majority, because that means that 47 species are still completely unique to each data set, which is not ideal. A plurality of the TELSeq specific species are found in only one of their technical replicates, but the other ones were found multiple times, so that gives me a little bit more hope. However, almost all of the Illumina specific species were only found in one technical replicate. I picked 7 other species that were found in 7 species to sort of like do a bit of a deep dive comparison between Illumina versus TELSeq, and I tried to pick the ones where there were multiple mags discovered in the Illumina dataset. So in terms of quality, it looks like Illumina only and TELSeq only mags are about equivalent quality-wise. There doesn't seem to be much of a quality difference in terms of completeness, contamination, honestly, genome fraction, proportion of unaligned contigs. In terms of strain heterogeneity, asterisk, it looks like the TELSeq mags picked up on a little bit more strain heterogeneity, though this, again, there's really no consistent trend here. There was also just more sequencing material in the original TELSeq sample, so maybe that's why they picked up on more variation, but I mean, I think there's way too many steps to truly like isolate where that source of variation comes from. And now I'm gonna skip all the way to the end. So just as a recap of everything I've talked about, the Microbiome in a Bottle project has 3 goals. The first of which is to comprehensively profile a healthy human gut microbiome using multiple DNA extraction methods, technical replicates, and sequencing technology. The second is to design a multi-technology integration workflow that can actually utilize to the fullest extent all of the data that I've generated. And the third is to curate a set of high confidence isolate genomes that fully comprise a healthy human gut. So in terms of progress, I've made all my data sets already that I will be working on in phase 1 of Microbiome in a Bottle. Phase 2 will hopefully include PacBio, hi-fi, high quality long reads. in terms of designing the integration workflow, I have developed the CAMP, a core analysis modular pipeline for short read analysis. And I'm currently in the process of developing the integration workflow, and that has sort of been also work in progress as well. Because I think the best way to create something that's useful is to wait for the problems to come up with first. It's great to anticipate them, but then that leaves you sometimes with tools that are designed without actually having a data set to work on. In terms of the high confidence isolate genomes, I have made all of my first pass initial draft mags, and the high confidence final draft isolate genomes are on the way. So takeaways plus microbiome in a bottle is that the challenges that I mentioned earlier, we've tried to address them with the microbiome in a bottle data set. It's not purely for pangenomics, but I think it's really applicable. Because metagenomics data is so complex and contingent, we just made a lot of sequencing data, like a lot of sequencing data. And hopefully the pairing of that, all the different types of sequencing technologies, plus all of the technical replicates, as well as the high confidence final draft isolates, will serve as like an answer key when you want to compare the starting point and the end point. And hopefully we have at least sort of made the resolving the species level genomes and the manual curation a little bit easier with the CAMP workflow that I've referred to a few times. So where to from here? Pangenomics from metagenomics is sort of, I think, like phase 3 of the microbiome in a bottle project. We've got the data, we got the workflow, we've made the high confidence final draft isolate answer key. Where do we go from here? We of course we dig further in. We look. Hopefully, we want to be able to look at strains. We want to be able to investigate all of the subpopulation level variation in a human gut microbiome. We want to be able to look at highly divergent regions in bacterial genomes that exhibit significant variability. We want to be able to identify genotypes that lead to phenotypes of interest, and we want to be able to even more maximally integrate all the sequencing data that we've generated. It would be, I agree, pretty difficult to identify necessarily mobile gene elements that are like fully extra chromosomal, but it would be super fun to find elements like recombination hotspots, like CRISPR-Cas loci, by like pathogenic islands by reassembling pangenomes from metagenomic reads. So we've got a data set, we've got a workflow, we are building the answer key right now. Let me know if any of this is something that you want to work on or that you have more questions about. So with that, I would like to thank my lab, the Hajira Suliha lab, as well as all the people I've worked with over the years. The Microbiome in a Bottle project is very much the child that takes a village to raise it. And luckily for me, I've got a really great village to help me raise it. So thank you all as well for being here. Thank you to Simone and Paola and Gianluca for inviting me, and I'm happy to take questions.
Thank you very much, Lauren, for this lovely talk, and congratulations for this terrific job. I mean, you made it clear how difficult it is, so congratulations on it as well. And yeah, I'm sorry, this is way above my knowledge on the topic, so forgive my questions. In the meantime, if the audience has some questions, please don't be afraid to ask. I will start with mine, which are, I think, more of like curiosity. So about these mags you were describing, do you know how typically how large they are and how similar are they across species?
Um, okay, right. Okay, that's a fantastic question. I unfortunately did not have time to go into the size of different mags. So, this little factoid is actually not super easy to dig up. Like And I remembered, like, I think a few years ago, trying to figure out what the average size of a bacterial genome is, because that's sort of like the size that you should be aiming for to reconstruct mags, right? If you have a mag that's 500 kilo base pairs, you know it's too small. If you have one that's 10 and you know it's too big, you should be able to bound it. So the average bacterial genome is around 3 megabase pairs in size. That was I had to dig really far back in the literature to find that. And most of my mags are about like 2 to 3, so definitely smaller than as an expected bacterial genome. But that's actually for a very, very well characterized reason, in the sense that it's very hard to actually bin rRNA, tRNA genes, and that is the content that most mags will miss, because they are so well conserved and so difficult to uniquely assign to a single mag. So you're going to miss like thousands of base pairs of material just because you cannot specifically assign it to a single cluster, and they don't have those species specific signatures of tetranucleotide frequencies. Um, and what is also an issue, in terms of like not necessarily the mag creation step, but downstream, is that a lot of mags will be deeply incomplete, and they will still be very similar to existing species in reference databases, um, but it'll be harder to calculate average nucleotide identity, because it really depends on how you calculate average nucleotide identity. If you just do a MASH signature, it might show up as very similar. But if you have something that's alignment based, that really depends on the quality of your assembly, then the less you have aligned, the worse your ANI will be. Even if it's actually very similar, you just can't align your material properly. So, very long way to say the size of the mag sometimes will determine the quality and sometimes will not, depending on your initial sequencing data.
Okay, okay, get it. Thank you very much. Uh, Last one that I have is, um, from what I understand, this is still a work in progress. I know
Mm-hmm.
you are also working on these metasub projects,
Mm-hmm.
so the metagenomic sampling and
Mm-hmm.
analysis of subways around the world. And is any of this data, even the preliminary one, publicly available, or is it still all in work in progress in Cornell?
So the Metasub data is very much public. You can find it at geoseq.com, spelled geoseeq.com. So all of that, from like the raw sequencing reads to some of like the intermediate short read classification files, can be downloaded from there. The microbiome bottle in a project is still a work in progress, though hopefully we hope to be able to release the phase 1 data at some point this year. But if anyone's interested in working with it or has any interesting project ideas, I very much encourage them to reach out to me, or reach out to Dr. Iman Hadria Zuliha, who's my supervisor, so we can talk about ideas together.
Okay, great. Thank you very much, Lauren. Thank you for participating and for this beautiful talk. Um,
Yes.
yeah,
Thank
I
you for
think
having
that's
me.
it. Thank you very much,
Thank
and
you.
see you soon.
Yeah, see you soon. Bye.